first speaker, who is Haines from the University of Texas, Austin, who will be presenting a talk entitled Spine Apparatus Distribution and Related Synapse Morphology in Rat Hippocampal Dentate Gyrus Area CA1.
Okay, so my name is Haines, and I will be presenting on the spine apparatus distribution and the related synapse morphology in rat hippocampal area CA1 and dentate gyrus. So I'd like to start out with a review of the case of the patient HM. He was a young man who suffered from severe epileptic seizures, and to try and fix this, doctors went in and removed a large part of his medial temporal lobe, and this had the unintended consequence of him not being able to form new memories after the surgery. And so what was later found on was that they actually removed an area of the brain known as the hippocampus, and this structure is critical for learning and memory. And so what I'm interested in is understanding the structural mechanisms that may underpin these information processes. So here is a sagittal view of the rat brain, and so that's just cut with this orientation. And here is the region of interest, the hippocampus. Now within the hippocampus, I am specifically interested in two regions, area CA1 and the dentate gyrus. So the reason I'm interested in these two specific regions is that they're distinct neuronal populations in area CA1, the pyramidal cell, and in the dentate gyrus, the granule cell. They receive similar presynaptic inputs from the entorhinal cortex. Specifically, the lateral layer 2, which is this, projects to the outer molecular layer of the dentate gyrus, and the medial portion of layer 2 projects to the middle molecular layer of the dentate gyrus, and that refers to the proximity to the cell body that the dendrites are located. And so then when looking at the area CA1 pyramidal neuron, both the lateral and medial layer 3 projections go to stratum oculus and moleculare within that cell. And so to get you acquainted with the structures that you'll be looking at today, this is an electron micrograph of the neurophil, and in yellow here is a dendrite, and then in green is an axon. And so information flow in the brain goes from presynaptic axon to postsynaptic dendrite. And dendrites have these occasional protrusions, which are known as dendritic spines. And dendritic spines are actually the location of most excitatory synapses in the brain. And a subset of spines contain the organelle smooth endoplasmic reticulum, as shown in this spine here. And the smooth endoplasmic reticulum can then go on to form an elaborated complex known as the spine apparatus, which is pictured here. So this led to the question of, for these two distinct cell types, how is SCR and the spine apparatus distributed along their dendritic arbors? And so to answer this, we took tissue from both of these regions, and we took it from the SLN in the CA1 pyramidal neuron, and then from the middle and outer molecular layer of the dentate gyrus. So once we have this tissue, it is then processed and stained for EM imaging, and then the tissue is embedded in plastic so that it can then be ultrasectioned to about 45 micrometers. And so you actually get a stack of sections, and if you take an image of each serial section, then what you can get is a stack of images that when you trace these structures, you can build three-dimensional models. And so that is the tools that we use to analyze this question. So for the dendrites in these three distinct layers, we took average caliber dendrites that is based on their microtubule numbers. And it has been shown that microtubule number is actually a good proxy for the relative size of the dendrites. And so in these 3D reconstructions, the dendrites are in pale yellow, the excitatory synapses are in red, the inhibitory synapses are in blue, and then the SCR, so a single SCR tubule, that's pictured in green, and then a elaborated SCR structure, the spine apparatus, will be in purple. And so these are the three categories for which SCR can form in dendritic spines. So there can be no SCR, there can be the single tubule, and then there can be the spine apparatus. And what's important about the SCR is that it functions in the storage and release of calcium. It also is part of protein modification after translation, and it also functions in lipid synthesis. So it's thought to be a good candidate for the regulation of synaptic plasticity or changes in synapse structure. So then we also categorize, so the two categories of the synapses are excitatory and inhibitory. And excitatory synapses tend to stimulate a response in the postsynaptic cell, while inhibitories tend to inhibit a response in the cell. So when characterizing the synapse morphology in these three layers, what we first had to do was look at the distribution of synapses across the layers. So we looked at the density in the MML, OML, and SLM, and it was found that SLM dendrites had a significantly lower proportion or lower density of synapses along their lengths. However, interestingly, when looking at the percentage of all synapses across the three layers, it was found that SLM dendrites had a higher proportion of inhibitory synapses than the middle or outer molecular layers of the dentate gyrus. And once we understood the synapse distribution, we also had to take into account the spine distribution. And so again, we did a similar analysis where we looked at spine density per micron across the three layers, with N being the number of dendrites. And SLM dendrites, again, had a significantly lower number of spines. And that can actually be visualized in these pictures above for the reconstructions. These have many more protrusions than the SLM dendrite. And then once we knew the frequency of the spines in the three layers, we then went on to look at what those spines contain. And so the percentage of spines for all the, in each of the layers, the SLM dendrites had a significantly higher proportion of spines that contained some type of SER structure. So what we then did is looked at, so we know that the SLM dendrites have fewer synapses, but they have more inhibitory, and then they also have fewer spines, but they contain more SER structures. So then we plotted the SER for each SER category. We wanted to know the corresponding synapse morphology characterized by synapse size in each of the three layers. And so the first result that was found from this is that SLM spines, no matter the SER category, had a significantly larger synapse size than the middle or outer molecular layers. It was also found that within each layer, the spines that contained a spine apparatus had a significantly larger synapse than the spines that had only an SCR tubule or no SCR at all. And then the last result that we found from this analysis was that the SLN dendrites that had a spine apparatus had significantly larger synapses than all other spine categories, by layer and by SCR category. So when looking at all of this data taken together, we were able to take some conclusions, and those were that the distribution of the spine apparatus tends to be differentiated by cell type, so the granule cell versus the pyramidal neuron. And it also seems to be a function of the caliber of the dendrite and the proximity to the cell body.
So you're at 12 minutes with that conclusion.
Okay. And so then the take-home message from this is that the differential patterns of the spine apparatus distribution in the area CA1 and the dentate gyrus, that this differentiation may serve to integrate the synaptic inputs from the entorhinal cortex and could play a role in synaptic plasticity and how the signals are actually integrated for learning and memory. So I'd like to thank you all for having me, and any questions? Yes.
How do you define the inhibitory synapses versus the excitatory synapses during the...
Oh, okay. So the inhibitory synapses are usually found on the shaft of the dendrite, whereas the excitatory synapses are found on the usually on dendritic spines. And also, because of the staining processes that the tissue has to go through, the inhibitory synapses, which usually contain the neurotransmitter GABA, that's actually the vesicles become distorted during the processing, and so those synapses that are inhibitory tend to have pleomorphic vesicles, so whereas the excitatory synapses have usually round and it's uniform. So, and that's how we're able to tell that difference.
One more question.
Where do you go from here in this? Like, what do you have any applications you hope this ends up being something?
So going from here, so I want to look at, so within the pyramidal neuron, we looked at the how the spine apparatus distribution changes as a function of proximity to the cell body and the dentate granule cell, but we still need to do that in the pyramidal neuron, and so going in and looking at a different layer of that cell type, then we can really do an analysis of whether it is the distribution of those SR structures is controlled by the presynaptic input or the postsynaptic cell type. And so that's really what we're hoping to get, is knowing